Okay, so you guys kindly invited me to talk about this paper, and I'm so happy that you did. This paper was published, as was mentioned by Leon earlier. No, not earlier this year, last year, and it really, I think, shows that there's technology that remains. That technology remains a strong driving force here, and there's this interplay here between questions and technology. And one thing that's driven the technology is just this realization. And you may agree, you may disagree. I'd be really interested to know what you think. But I think what we've witnessed is this recognition that the transcriptome is an amazing proxy for what's going on in the cell. That was always something that back in 20 years ago when I started working on the transcriptome, whenever I would show my microarray data, people always say, yeah, but transcripts don't matter, it's the proteins. That's what the comment I would always get. And I would have my set of responses like, there's no protein without a transcript, as far as I know, and things like that. But it was always on us to validate things at the protein level. And what we've seen is a growing recognition that estimating the cell as a bag of RNA is actually a very, very useful proxy for function. I'd be interested to know what you think. It could be, of course, also that we're looking under the lamppost, right? Because it's become very easy for us to measure what the cell is doing in terms of RNA. And the proteomics, even though proteomics has made amazing strides, and we saw a big paper in Nature yesterday about the progress in proteomics, and Hanno Steen is here representing the proteomics world as well too, and I'm sure others. But for some reason, transcriptomics has been a couple decades ahead, and this has led to a lot of elucidation of many problems using this technique. So what I want to tell you about is, first of all, the people who did this work. The paper was led here by Ruben, and he now actually has graduated, and he works with Aviv Regev actually at Genentech. So he moved to San Francisco. But I'll also tell you about work from Maayan and Dalia in the lab. And unfortunately, about the work of others, I won't get a chance so much to mention, except for a review by Anjali. Okay, so what I want to tell you is 6 things. But again, if we don't get to everything, it's fine. I'm much more interested to know what you guys are interested in, and we can stop anytime. I want to tell you about the topic is cancer. We're using spatial transcriptomics and single-cell RNA-seq to learn about tumor heterogeneity. And I'll tell you, I'll introduce this topic to you first, and then I'll introduce spatial transcriptomics as a broad field, but also specifically this method of array-based transcriptomics. I'll tell you about a method, and this is the method that we described in the paper. And then since the paper, we have done more work. The paper was published 1.5 years ago, so in a sense, in our lab, it's considered ancient history. So I'll tell you about what's going on right now in terms of further integrating single-cell RNA-seq to define cancer cell states, and then mapping those single-cell states to their location in the tumor using spatial transcriptomics to understand the connections between cell states and the TME, the tumor microenvironment. And in general, what I want to say is that spatial transcriptomics is one of these new technologies, just like the human genome was, in a sense, a kind of totally new sort of data set, or just like electronic health records are a totally new sort of data set. Spatial transcriptomics also produce a new kind of data set, and it opens up possibilities beyond hypothesis testing, but also into exploratory. And this is something that we've learned also from single-cell RNA-seq, that the data is so vast, you can look at it as an atlas where you can find many, many new directions. So I'll tell you about our thinking of this. Okay, so in general, and I'm not, feel free to put your questions in the chat, but if Jeff and Leon can help me voice those questions, because I won't be monitoring it, but just bring it up and we'll start it up. How's it going so far? Any questions so far? So far, so good? All right, let's jump right in. So I think I don't need to tell this crowd, this is probably obvious to all of you, but single-cell RNA-seq is, in a sense, given a rigorous definition of what a cell type and what a cell state is. And that's something very novel. Before single-cell RNA-seq, we thought about cells and cell types in terms of their morphology and function. But now we have a molecular signature of what a cell type is. Now, when we see this cloud of points where each point is a cell, and we notice it's not really a cloud if you think about it, it's a set of clouds where each little cloud or cluster, if you will, really turns out to be a cell type. So now we see that, first of all, let's acknowledge it didn't have to be that way. It didn't have to be that if we measure the transcriptomes, they would cluster so discretely. And the fact that they do was I would say, unexpected in some respects, and has now been used as the defining feature of what a cell type is. And there are, this has led to a huge amount of work in the field. There are so many people working on exactly how do you define what a cell type is now? How do you use these signatures? What is a cell type? What are the relationships? And I think it's now taking this old question of what is a cell type and making it very modern and quantitative and rigorous with this technology. So that's very exciting. With regards to cancer, we have also seen that when you take a tumor and you study with single-cell RNA-seq just the cancer cells, you see heterogeneity. You see that the cancer cells are not all of the same transcriptome, but rather are in distinct states. So for example, in gliomas, we see that there's a stem-like state. Some of the cancer cells are in a stem-like state. Some are in an astro-like state. Some are in an oligodendrocyte-like state. And if you look at, I don't know, colon cancer also, for example, you have the enterocyte-like state. You have a goblet-like state. You have a progenitor-like state. So the cancer cells are heterogeneous in terms of their states. That was striking to me when I first saw it. We saw it in melanoma when we take the cancer cells. Right, so imagine, like in the previous slide, you have all your cell types. We take just the cancer cells, now do a dimensional reduction analysis. This is a PCA on just the cancer cells, and they form, not completely discrete, but there's very much the notion that you're capturing here structure in the data, that there are states. So one thing that caught our attention is that in this melanoma, there's a stress-like state. And we didn't know what to do about that. So I want to tell you a story about this state, because you could say, isn't that a complete artifact? Because if you think about how you make single-cell RNA-seq data, you first make a single-cell suspension, and then you start the pipeline that leads to their sequencing, right? Maybe during that stage of making a single-cell suspension, the cells are stressed out. So just in vitro, you're activating a new state. It's not endogenous to the tumor. That's a possibility. The thing is, though, we saw this state in many other tumors. So here you're seeing a PCA plot for different cancer types, and each circle is a cell, and we're coloring that cell in terms of how well it scores for being in the stress-like state. So here, this is melanoma, but it's a different tumor than the one I showed you in the previous slide, so it looks a little bit different, but still you could see that there's 3 states, and one state is the stress-like state. Here's an oligodendroglioma. These cells here are in the stress-like state, but you could say they just fall prey to the same mistake. In other words, we're making the same mistake each time because it's single-cell RNA-seq. This triple-negative breast cancer data was from nuclei sequencing, though, so we think it's not that, and we use some other bioinformatics controls, but Ultimately, though, the single cell RNA-seq didn't allow us to rule out the possibility that it could be just an artifact. Okay, let's put that aside in a second. I'm going to come back to it in about 5 slides, but I want to tell you more generally, we have some questions about cell states. If the cancer cells are present in different state, you could ask, what is the source of that? How do they arise? Is it because of genetic differences? Maybe they're each different clones. Is it stochastic? Is it epigenetic? Is it signaling, perhaps because of the microenvironment? And let's say there are different states. Of what consequence is that? What does it mean? Maybe you can measure it, but it doesn't mean anything. Or alternatively, there's the hypothesis that actually it is important. Perhaps it leads to the cooperation among the cancer cells. So there are many questions of what does it mean? Is it important? How do we tackle that? So in this slide, my point will be that spatial transcriptomics is a great way to tackle that. And Bo Xia made this presentation. Actually, I have to tell you, Bo has been doing extraordinary things lately. And just today, there's an article in the Times about his project in our lab that he led on something totally different, but maybe in the Q&A session, we'll talk about that. So what Bo is showing here, is Legos as individual cells. Okay, so the analogy here is that each Lego piece is a cell. And when you're doing bulk, of course, it's a big mishmash, it's a mix. You're not sure how to interpret the transcriptome. It's very difficult, even though that's what we were doing for years. But with single cell RNA-seq, you're getting the clusters. So now you can tease out the expression profile of each cell type. And that's great. The problem is, or one of the problems, is that you've lost the spatial dimension, obviously, because the first step was to make a single cell suspension. So you don't know how it all fits together. You don't know how they're interacting. You don't get any possible clues about what a state means from their neighbors. And so spatial transcriptomics is meant to address that. Now, Jeff is a pioneer of spatial transcriptomics, and I'll mention his work in the next slide. But on this slide, I'm going to tell you about a spatial transcriptomics that's different from his approach. This one is now marketed by a company. I assure you, I am not getting any percentages, anything, although as much as I'd like to. But the company is called 10x Genome. It's the same company that's been very popular for single cell RNA-seq, and the product is called Visium. This is probably completely old news to 99% of you in the audience. Every array that you buy a slide, there's 4 arrays. Each array has 5,000 spots. And then at the end of the day or 2 days, you get a gene expression matrix. Okay, so this gene expression matrix is of the same style as the gene expression matrix that you get for single cell RNA-seq. The rows are genes, but the columns, as opposed to being cells like in single cell RNA-seq, each column gives you the transcriptome for a particular spot. So what I should show you here, I don't know why I don't, now that I think about it, is that there's actually 2 more vectors underneath here that for every spot give you the X and Y coordinates for each column. I'm going to change that in the next time I present this. And now, as I mentioned, this is just one way of doing spatial transcriptomics. Okay, in a review that we published last month, we talk about the full range of spatial transcriptomics. Let me just point out that from my perspective, I'll be interested to know if Jeff agrees, the essential output of all of these techniques. So before I talk about their differences, I just want to talk about their similarities. The essential output is the gene expression matrix. Okay. Oh, here I do show the XY coordinates. You see, for every spot, you see the XY coordinates. And what the other methods do that's different is is something that gives the advantage, but perhaps also a slight disadvantage. One amazing advantage is that other methods are actually using imaging, and so they're at the single cell level. right? So here, the gene expression matrix actually gives you the transcriptome for in an individual cell, whereas the spatial transcriptomics that's sold by Visium is not at the single cell level yet. It's like about 5 to 10 cells that are on top of each spot, okay? I should say that there's a new product coming out in a few months, and I'm told it's essentially at the single cell level, but that's not out yet. You can't build on it. The potential disadvantage of these imaging-based methods is that at least yet, it's not omic. It doesn't give you the measurement of every single gene. Now, these NGS-based methods, the next generation sequencing-based methods, are also, strictly speaking, not whole genome, because they're all premised on the notion that they're going to capture polyadenylated transcripts. So microRNAs are not polyadenylated. Link RNAs are polyadenylated. Some histones, some histone genes are not polyadenylated, so you won't get those. And so what else is not polyadenylated? Ribosomal RNA, transfer RNA. So it's strictly speaking not in omic, but it's essentially omic. Okay, now, can we use this method to query for our stress-like state? Remember, we found this state and we're not sure if it's an artifact. Well, with spatial transcriptomics, you can avert the possibility of an artifact, because what you do is you take the tissue and freeze it right away. So there's no possibility of a new state arising in vitro or post surgery. And here you're looking at a melanoma sample, and we are querying for the gene expression of genes that are in the stress-like signature, and we observe them. You can see FOS here is higher in the tumor than outside the tumor. So we could use spatial transcriptomics to validate our states that were identified with the single-cell RNA-seq. So in our minds, this is great. This is like, oh, wow, the two can work really well together, single-cell RNA-seq and spatial transcriptomics, because at least during the time that we were working on this paper a couple of years ago, the two techniques had blind sides that we thought that each one could complement the blind side of the other. In other words, we saw them as mutually illuminating. Why? Because the single cell RNA-seq is at the individual cell level, but lacks spatial resolution. The spatial transcriptomics has spatial resolution, but was not at the single cell level. So we thought, oh, wouldn't it be great if for a given sample, you divide it in half, one half you send for single cell RNA-seq, and the other half you do spatial transcriptomics, and then you would integrate it at the analysis stage. So that was the premise of the paper that you so kindly invited me here to speak about. Here are two tumors. This one here, labeled PDAC-A. So this is pancreatic ductal adenocarcinoma. At NYU, we collaborated with surgeons, and approximately 60 minutes post-surgery, we get the tumor, we put it on ice, we freeze it down right away. And then with the help of a pathologist, we can annotate the regions. This red area is annotated as cancer. Here we have ductal epithelium. This is normal. This was annotated by the pathologist as normal, and so forth. And now, remember, for every spot here, we also have the transcript. Oh, you can't see the spots, but you can imagine that there's spots underneath here. And this was before the time when Visium was around. So we would buy the slides from a company called Spatial Transcriptomics. And back then, it wasn't 5,000 spot, it was 1,000 spots. So that's the resolution that you're looking at here for this paper. And if you now do principal components analysis on the transcriptomes, and now map the scores, because for each transcriptome, you also have the XY coordinate, you can see that the transcriptomics, even though it doesn't know anything about the pathologist, the transcriptomic, the PCA, is just looking at the gene expression matrix, It doesn't know what the pathologist wrote here, but still the transcriptomics recovers the central features. You see it identifies this normal epithelium region, it identified this region, and so forth. And similar with another sample. But now we ask, how would you integrate it? And here's the method we came up with. I think today we might do it a little bit differently, but before we talk about that, let me just, I guess everybody always looks back on their work 2 years ago and says, oh, maybe we would have done it a little bit differently now. But regardless, let me explain to you what we did in the paper. So we said, okay, we have the single cell data. And just to remind you, it looks like this. This is the single cell data from a PDAC sample. And so we could identify, these are the red blood cells, these are the acinar. We have the gene expression profile for each cell type. Okay, so for example, for fibroblasts here, we have some fibroblast cells and we have their transcriptome. We can identify which genes are specifically expressed by each cell type, right? Because we have the gene expression data. Also, we can identify different regions, and we can say which regions are differentially expressed in each region. So for example, we could take this region and say, okay, which genes are much higher in their expression here than here? And we can make lists. And the whole notion of how we would integrate these two technologies relies on a comparison of lists. So if you think I'm about to give you some heavy duty math and equations, you're going to be pleasantly surprised. This is actually super simple. It's just comparing lists. For example, let's say we're asking, in this cancer region over here, which cell types are present in this region? So what we do is we take the genes, we have a list of 660 genes that we identified as being highly expressed here. Now we look at the single cell data and we say, fibroblasts, let's ask, are fibroblasts there? And we have a list of 555 genes that are high in the fibroblasts. We compare these lists and we see that 58 genes are present in both lists, okay? And that's out of about 20,000 genes that we studied. That's the power of this method. These two methods is that they're omic, they're all of the genes. And so now we can use the hypergeometric distribution, plug in these four numbers, and it tells us how surprised we are to see such an overlap. Is it much more than we would have expected randomly? Much less. And in this case, the p-value is 0.0000014 zeros, and then a 1444. So it's a very low p-value, which provides evidence that it's actually, statistically speaking, there are a lot more fibroblast-specific genes expressed in this area than you would expect. So from this, we would conclude that, okay, we're detecting fibroblasts in this cancer region. So this is just a simple computation. Imagine now taking this computation and running wild with it and systematically doing it for all the possible combinations, all the areas versus all of the cell types. And if you do that, you get what we call the MIA map, which is multimodal intersection analysis. The computation we just went through in detail is this one. Every column corresponds to a region. Okay, so this is the region we call the cancer region. This is the pancreatic tissue or the normal region. And for each region, we can ask which cell types are enriched or also depleted. So for example, cancer cluster number 1 is depleted in the normal region, as actually you would have expected. Otherwise, it wouldn't be normal, it would be cancer. And so we thought this was really cool because for every region, you could deconvolve it into the resident cell types. Now, let me tell you, this is something that I think if we ran today, it would be of much higher quality. And the reason was that we generated this data in 2016. And the technology was not as good as it is today, let's say that gently. And we were just happy that it works at all. And what you do when the technology is very new and the data is sort of rough, you do the bioinformatics trick of averaging things out, right? When the data is not good, there's sort of like a bioinformatics principle that you just bin things more and more, right? The less you have to bin, the higher resolution your data set is, I would venture. I'd be interested in your thoughts on that. And so here you can see we created 4 regions, but today we would be able to run this on a spot by spot basis because the data is just over 10 times better. So, however, at the single cell level, we didn't have to do so much averaging. You remember in the analysis I just told you about, the unit of analysis was a cell type, but at the single cell RNA-seq level, you can also identify different states. So here, if you look at just the ductal cells, you can see that it could be further classified into individual states. Like, why should we pull together all of the ductal cells when really there's so much variation? So we identified markers unique to each cell state. And it was also pretty novel to identify these states because previously in the literature, there were 2 states known that we had identified in 2016 in a different paper. And here we brought it up to 4. And now we were in this position where we identified states at the single cell RNA-seq level, but we don't know what they mean. And so the spatial transcriptomics could be a really good tool to help you figure out what it means.